Okay, today we're going to be talking about CRISPR-Cas9, which is a method used to mutate genes. And so what does it mean to mutate a gene? Well, it means that we alter the DNA sequence so the resulting protein is either no longer made or it's a non-functional version of the protein. And so this is also referred to as knocking out a gene. And so that's what we're going to learn about today. And why would you want to specifically use CRISPR-Cas9 to knock out a gene? Well, let's say you're a scientist and you're studying a particular trait or a gene in an organism. The kind of gold standard to test what it actually does is to knock it out. And so say you'd have a hypothesis about your gene. In this case, let's say your gene is called Fluffy. In order to test what it does, you'd want to knock it out and then see what happens to the resulting animals. So if our hypothesis is that Fluffy makes dogs fluffy, then we would expect that, so in our starting dog, it's of course fluffy because it has a normal version of the gene or wild type. And then after we have used CRISPR-Cas9 to knock out the gene, we would then expect that the resulting dog is no longer fluffy. And so this would be our fluffy mutant. But of course, you actually need to do the experiment to see whether or not this is true. And so we can learn something about the role of the fluffy gene in dogs. So how would we actually do this with CRISPR-Cas9? Well, there are two requirements. So first of all, in order to mutate a gene, you need to cut the DNA. And secondly, if you're going to cut the DNA, of course, you need to do it specifically in only the fluffy gene, not in another gene. Otherwise, that could kind of ruin your experiments. And so you need specificity. And using the CRISPR-Cas9 system, these two points are fulfilled by two different components of the pathway. So what cuts the DNA is a protein called Cas9, and what provides the specificity is something called the guide RNA, or gRNA for short. And so what would this actually look like? Well, we can't instantly do the experiment in dogs, so we'd start with cells in culture. So let's say we have a Petri dish that has a lawn of cells on top of it. So we need to zoom in to one of those cells. And then of course the DNA is actually inside the nucleus, so let's zoom in on a specific part of the nucleus to the region of DNA that encodes the fluffy gene. And so when we're using CRISPR-Cas9, as I mentioned, the guide RNA will provide the specificity. And so here is our fluffy gene. Here is the Cas9, which I told you is the component that actually cuts the DNA. And here is the guide RNA, which provides the specificity. And so this guide RNA is going to home in on the correct part of the DNA so that the mutation can be created. So what actually happens when the guide RNA takes Cas9 to the DNA locus? Well, the DNA unfolds a little bit so that it's now single-stranded in one part. And Cas9 will come around the DNA, and the guide RNA will bind to the DNA locus. And now, as we mentioned, Cas9 is a protein, it's an enzyme, so it's able to actually cut the DNA. So it has these kind of molecular pincers that will cut it. And what actually results from this is the DNA now has a double strand break. And so there are many different ways that DNA can be mutated, both sort of naturally from, for instance, UV radiation, or induced by something like Cas9. So sometimes it'll be just a single base pair change, or a single strand will be cut. But this is a specific kind of cut, which is a double strand break. And so what actually happens when the DNA has a double strand break? Well, because this is a sort of unnatural event for the DNA to happen, this is going to set off a kind of alarm bells in the cell, and it's going to try and repair it. And it uses a pathway that's called non-homologous end joining. A bit of a mouthful, you can call it NHEJ. And so it's good that the cell has this pathway to repair the DNA, but there's an issue, which is that the cell doesn't actually know whether any DNA was removed from that site or added, and so it has to repair it without knowing what actually happened. And so when it repairs that DNA, it often will either insert or remove a couple of base pairs. And so this is called an insertion or deletion, or an indel. And so what's important about CRISPR-Cas9 is that the mutation itself is not actually caused by the fact that you cut the DNA, because the DNA is repaired. The mutation, and so the resulting knockout, is caused by the fact that there's an insertion or removal of DNA base pairs here. And I'll show you why. So let's say that we have a sequence ATG, CC, AT, C, T, A, G. And so, as you may know, ATG is a start codon, and the rest of these will encode the protein. So let's say that after our CRISPR-Cas9, we've had an insertion of one base pair. So now this will read slightly differently because there's an added A. And so that alone doesn't seem like a particularly big deal for the cell, just one extra A. But of course, the way that the DNA is read into, or the RNA is read into protein, is with codons, which are a series of 3 nucleotides that are converted into amino acids. So in this case, this just makes a number of different amino acids that will keep going throughout the whole protein. Now that we've inserted a nucleotide, this changes what amino acids are made in what's called a frame shift. And in this particular case, now that CRISPR-Cas9 has inserted this A, or as a result of CRISPR-Cas9 an A has been inserted, we now have this important codon here, which is actually a stop codon, which tells the ribosome to stop translating. And so this original gene and the resulting protein would have a number of amino acids, maybe 200 amino acids. But now, given that this is the start, and 3 amino acids later we have a stop, we now have a truncated protein. And so let's say that this was the normal fluffy gene. This is now the fluffy protein that's made from the fluffy gene. As a result of our mutation, we now have a really short, tiny, fluffy protein. As a result of CRISPR-Cas9. And so the idea is that this protein would not be functional because it doesn't have the full sequence, and it would probably actually be degraded in the cell. So let's now bring this back to our original hypothesis. So we thought that if we knocked out the fluffy gene, we might get dogs that are no longer fluffy. And so as a result of those experiments, we now have a mutation in the fluffy gene. And so if we were to actually have done this in dogs, the first time you mutate the DNA, you're only going to mutate one copy of it. So this will be a heterozygous dog. And so in order to test our hypothesis, we need a homozygous, so there's no fluffy protein made at all. So we need to cross this heterozygous dog with another heterozygous dog. And now we'd look at the resulting progeny. So we'd expect that 25% of them would be wild type, normal dogs. And so they should look fluffy as normal. Then we'd expect that 50% of them would be heterozygous dogs. And so just like their parents, they would also be fluffy. And then this is where our hypothesis would be tested, because we'd be interested in looking at the homozygous ones. And so if our hypothesis was correct, 25% of these dogs should now be fluffyless and maybe a little bit sad.